Hello, everyone, and welcome. The webinar will start in 5 minutes.
So we will wait 5 more minutes.
Yeah, but we're live now.
I know.
And to everyone who's already here, you can see how fresh Dr. Ahmet is right now in nature. One more minute and we'll start. And good morning, good afternoon, and good evening, everyone. Welcome to the online workshop on recent advancements in stem cell and exosome therapy for male infertility. This is episode 2 of our Stem Cells in IVF webinar series by the International Fertility Group. And you can access episode 1 on our YouTube channel. But for now, my name is Sophia and I will be your moderator for this session. Our 3 main objectives for this session are to discuss the following: a background on male infertility, how stem cells and specifically which types can be used to treat male infertility, and case studies demonstrating the application of stem cell and exosome therapy. I'm confident that this will be an engaging session, but not without your participation. So please feel free to ask questions via the chat box, and they will be answered after the presentation. We will also share some upcoming event details at the end, which will benefit all of you who are interested in advancing your practice in stem cell therapies. So stay tuned. And now, let me introduce you to our speaker, Dr. Ahmet Tevfik Albayrak, urologist as well as reproductive and sexual health expert. He's a distinguished member of the European Society for Sexual Medicine, a testament to his expertise and dedication in the field of sexual health. And for this reason, we are fortunate to have him share his expertise with us today. And without further ado, I'll be giving the floor to Dr. Ahmet.
Thank you, Sophia. Now, hello everybody, first of all. Now we can start the presentation. May I start? Yeah, Sophia, of course, right? Okay, I start. First thing first, yeah, background on male infertility. Okay, what is male infertility? Infertility is usually defined as the inability of couple to conceive even after 1 year of unprotected, frequent sexual intercourse. The male is solely responsible in about 20% of cases and is a contributing factor in another 30 to 40% of all infertility cases. It's much, actually. As male and female causes often coexist, it is essential that both partners are investigated for infertility and managed together. Overall, the male factor substantially contributes to about 50% of all cases of infertility. The other slide, please. The main causes of male infertility are the absence of sperm in the ejaculate, which means Azoospermia, or a decrease in sperm quantity, Oligospermia, or motility, Astenospermia, or poor sperm quality, Teratozospermia. These pathologies constitute up to 90% of the problems related to all male infertility. The other slide, please. These pathologies caused by endocrine disorders, usually due to hypogonadism, estimated at 2% to 5% of cases. Sperm transport disorders, which means obstruction or maybe a vasectomy history, at 5%, and primary testicular defects, which at 65% to 85%, actually. And an idiopathic one, it is like 10 to 20%. And sometimes cancer, up to 6% of men evaluated for male infertility will be found to have more serious underlying pathology, such as cancer. The other one. Okay, the initial evolution, it is important. This includes a detailed sexual history and physical examination, of course. Also, with 2 separate semen analyses, hormonal testing and an optional cervical ultrasound can then be performed if abnormalities are found. The key purpose for evaluating a male for infertility is to identify the contributing factors, offer treatments for those that are reversible, determine if the patient is a candidate for assisted reproductive techniques, and offer counseling for irreversible and untreatable conditions. Other slide, please. Okay, azoospermia. Azoospermia is defined as the absence of sperm in the ejaculate after centrifugation during semen analysis. Obstructive azoospermia accounts for 40% of all azoospermia cases. It has been reported that there are at least 2,000 genes related to spermatogenesis, and azoospermia accounts for 25% of the genetic causes of male infertility. This is important because in azoospermia, a Y dilation and chromosome analysis test should be done because Klinefelter syndrome is the most frequent chromosomal abnormality in patients with azoospermia. Recent studies have also revealed that microdeletion on the long arm of the Y chromosome, especially at the azoospermic factor ESF region, results in impaired spermatogenesis and are genetic cause of the most common sperm failure resulting in male infertility. The other slide, please. Oligospermia, astenospermia, teratospermia, and high sperm DNA fragmentation rates are the most common phenotypes of poor sperm quality. And varicocele with the etiology of intrinsic testicular impairment also induces testicular spermatogenesis by damaging sperm DNA. Factors such as infection, oxidative stress, smoking, and unhealthy lifestyle can also result in a poor sperm quality. The other slide, please. Okay, the main events, stem cells. In mammals, there are two types, embryonic and adults. Embryonic stem cells from the inner cell mass of the blastocyst, and adult stem cells such as bone marrow, adipose tissue, umbilical cord, and induced pluripotent stem cells. The other slide, please. Embryonic stem cells are from the inner cell mass of blastocysts, as we talked before. They're pluripotent by nature, and they can transform to the other structural cells. This is important. Keep in mind. We will talk it about later. The other slide, please. Okay, stem cell therapies. Stem cells have become a promising treatment options for infertility due to their numerous remarkable characteristics such as a multi-directional differentiation, angiogenesis, immune regulation, and paracrine stimulation. The proposed mechanism of stem cells in the repair of reproductive dysfunction includes the following steps. Stem cells migrate to the injured reproductive tissues caused by chemokines and then differentiate the integral somatic cells with known tumorigenic properties, of course. They may reside in the reproductive tissue and help ameliorate the damaged microenvironment by producing paracrine factors. Stem cells transplantation can also restore the endocrine function by secreting anti-inflammatory factors or increasing the number of T cell population, leading to immune suppression. Other slide, please. Okay, but what kind of stem cells? This is important. Adult stem cells actually can isolate from variety of tissues, including bone marrow, adipose tissue, Barton jellies, umbilical cord blood, human amniotic fluid, and even peripheral blood. Current reports have proposed that these stem cells could treat unexplained infertility. Mesenchymal stem cells are the most common adult stem cells, have the highest differentiation potential. However, spermatogonial stem cells may be more potent in case of infertility. Other slide, please. Okay, adipose-derived stem cells, they are most used stem cells because they are really easy to harvest, first of all. Adipose-derived stem cells already have been successfully used in the clinical treatment of traumatic defects, Berger disease, and cartilage bone regeneration. Adipose-derived stem cells seems to improve vascular remodeling and tissue recovery, indirectly through the secretion of paracrine cytokines or directly by differentiating into the injured cells. They have been used to address reproductive problems in animal models on infertility. For instance, here's a study. In the study from 2013, their male rats were sterilized with busulfan, with a chemotherapeutical factor. After confirmed infertile status by analyzing the testes of rats, the right testis of each rat was injected with stem cells, which labeled with GF protein. The other testis was left as control. After 12 weeks, testes of 8 animals were removed from analysis. The remaining male rats were mated with female rats. Cells from offspring were analyzed for GF protein expression. This is a very interesting study, by the way. The difference in dimension of stem cell injected testes and control testes were analyzed, and mass increase of almost 50% was measured in stem cells injected testes, which is remarkable. Moreover, the control testes were scanned histologically for spermatogenesis. It showed atrophy, complete and incomplete spermatocystic arrest, and Sertoli cell only appearance. However, the presence of spermatogonium in the other testes with stem cell transplantation was noted. Furthermore, the seminiferous tubules of the controls, which were not treated with stem cells, were empty and indicated the disruption of spermatogenesis, as I mentioned before. On the other hand, the tubules appeared to be filled up with spermatogenic cells in the sections of stem cells treated testes, but with low rate, but still. Also, spermatozoa were observed. Most interesting part of this study, these stem cell injected male rats were mated with other females, and preserved fertility was observed by obtaining the next offspring. To determine whether the offspring were originated from stem cell treated male spermatozoa, all blood from each animal were collected for DNA isolation and detection of GF protein gene in genome. GFP gene was detected. As you can see, the infertility was reversed in the study, actually. The other slide, please. And this spermatogonial stem cells, they are most promising because you can collect them from the testicular tissue, and it's very important. Responsible of male fertility because they are origin of spermatogenesis, ability of self-renewal and generation of large number of haploid cells, ability to convert into pluripotent cells without gene manipulations. For instance, for men who harbor spermatogonial stem cells, despite azoospermia, a testis biopsy combined with in vitro proliferation and transplantation may solve the problem. The other slide, please. Here's a spermatogonial stem cell study. In the study from 2020, the authors found that transplanted allogenic spermatogonial stem cells regenerate spermatogenesis and produce functional sperms. The other slide, please. There are multiple studies, as you can see, and reviews that have applied different stem cells to infertility model using the same study protocol. And the results of all of them are the same as the study we just talked about. Okay, now stem cells, but we should talk about exosomes too, because exosomes are the new stars, you know. Exosomes are the mainly disc-shaped extracellular vesicles with a diameter of 30 to 150 nanometers, secreted by living cells and have a phospholipid structure, which is not much different from the cell membrane structure. Exosomes exosomes contain a large number of protein, lipids, microRNAs, long non-coding RNAs, and so on. Exosomes exert their biological functions mainly in two ways. One is by transmitting communication from cell to cell, such as antigen presentation, which is the main way they perform their function. The other way, by removing excess or harmful molecules, which provides the possibility to study biomarkers. The other slide, please. Okay, at present, in-depth research on exosome mainly focused on stem cell therapy, cartilage repair, tumor metastasis, and organ development. This is this is especially true in the process of testicular development. Exosomes from testicular somatic cells and seminal plasma play an important role in spermatogonial. Actually, you can obtain exosomes from the lots of tissues. But if you want to treat infertility, you should obtain from the testicular tissue. Okay, this is important. Exosomes and testicular development. For instance, umbilical amniotic fluids, mesenchymal stem cells, and Sertoli cell-derived exosomes are found to reduce reactive oxygen species production and their effects on sperm DNA. Therefore, mesenchymal stem cell-derived exosomes can potentially improve stem cell quality and adhesion properties, as I said the same before, actually. The next, please. There are lots of exosomes from different cell sources, although adipose-derived exosomes are the most commonly used. Sertoli cells-derived exosomes are thought to be the most useful in the treatment of male infertility, as I said before. Okay, here's a study. It's a different and interesting study too. In the study, the authors use amniotic fluids derived exosomes for the restoration of injured testicular tissue and spermatogenesis in non-obstructive azoospermia bred induced by busulfan, like the other study. When we look at the results, a single injection of 40 microgram of exosome or a double injection of 10 microgram of exosome restored the sperm concentration, sperm motility, and the number of spermatogonia, spermatocytes, and elongated spermatids to the same levels of the control non-obstructive azoospermia rats. This is interesting, actually. Of course, more studies should be done on exosome therapy and male infertility, but for now, the results coming from the scientific community are promising. The other slide, please. Okay, and now take-home lessons. Considering the advantage of pluripotent capacity, stem cell therapies are considered as an alternative approach, not the main approach, alternative approach to assisted reproductive techniques to improve fertility treatment outcomes in humans. Mesenchymal stem cells predominantly play an instrumental role in the immunoregulatory function and the recovery of failing reproductive organs. On the other hand, stem cell therapies remain largely in the preclinical investigational phase, and several ethical challenges created by new scientific development in the field have aroused widespread conflicting opinions. The other, please. Stem cell therapy can be safely applied to all age groups. However, some studies argue that use of stem cells should be restricted in patients with a history of malignancy. If exosome therapy will be used in the treatment of male infertility, it is recommended to use Sertoli cells-derived exosomes. We have two more slides, but these slides are bonus slides. This is the slides show how can you the application of the stem cells or the exosomes, as you can see, you should use the best way to apply stem cells and exosome is the intratesticular injection, preferably to the testis, as you can see there. You can see from the ultrasound, and then you can use your needle and just inject it to the testis. The other slide, I just showed the anesthesia. Spermatic cord blockage will be fine. You can use it safely, no pain, don't worry, and use your stem cell. Thank you.
All right. Thank you, Dr. Ahmed, for that fascinating presentation. I'm sure we all learned a lot, but let's get right into the questions. And you are still welcome to ask your questions or send your messages on the chat box. But here are some questions so far. Let me just open them. We have two questions from Vilma Seth. And the first one is, does Zymot or ZYMOT really help if you have DNA fragmentation over 30%?
What really helped? Stem cells? I didn't hear.
Does Zymot, ZYMOT, really help if you have DNA fragmentation over 30%?
I couldn't understand exactly the question, but the stem cells application is really helped the DNA fragmentation. And 30% is not much, actually. You can use the stem cell application or the exosomes, but exosomes are not, I'm not sure, but the stem cells are definitely is helpful. But maybe Sertoli cells-derived exosomes may helpful too, but not adipose-derived or mesenchymal cells-derived exosomes.
Okay, the person asking has followed up and said that Zymot is a sperm sorting chip.
Sperm sorting chip?
Yeah. If it helps, if you have DNA fragmentation over 30%.
Actually, I don't know. I never heard about the diamond chips. Maybe you can write me down because I.
Okay, definitely. This person can always ask us via social media as well. I don't think this was discussed in the presentation, but the other question is, what is idea DNA fragmentation percent for natural conception?
Okay, if you have a DNA fragmentation, the DNA fragmentation, your sperms are gonna die basically. Actually, any DNA fragmentation because of the reactive chemicals or stress induced, most smoking, by the way, the sperms will be dead because their DNA will be fragmented, and it means dying.
Okay.
And varicocele is another. Actually, if you have varicocele, it is a big problem too for the DNA fragmentation, because no oxygen, just DNA fragmentation. If you smoke a lot, no oxygen, DNA fragmentation.
Okay. Now let's move on to the next questions. Are there any side effects or contraindications to such therapies?
Actually, if you have autoimmune diseases or systemic malignancies, the studies say you shouldn't do it. You shouldn't use it because it can make them, the worst. If you have autoimmune diseases or the malignancies, for now we don't use stem cells. This kind of patients.
Okay.
But no side effects for now. As we know, no side effects.
That sounds good. The next question: should we use stem cell therapy for all infertility patients?
Actually, no, no, no. If you, first of all, we must evaluate the patients, like if it's a hormonal or maybe physical, like varicocele or obstructive azoospermia. First, we need to find the reason. Then after that, we should cure the reason. But I always talk with my patients after varicocele therapy. I ask them, "Do you want stem cell therapy too? It make your sperms maybe better, but not 100%. But it's a supportive treatment, and it's not cheap. If you want, I can do it. But if you don't want it, it's all fine because you got a varicocele and we made the surgery. You will be well. But if you want it, we can use it."
Okay. And next stem, is a question of stem cell or exosome therapy. Which one for you is better?
Stem cell is better, but it's a harvesting uh, process. It's a process. Some patients don't want the harvesting process, so we just use the exosomes. But stem cells, for now, according to studies, are better than exosomes.
Okay. And I think our last question, does stem cell therapy efficiency decrease with age? And is there a limit, age limit?
Actually, there is no age limit according to studies. According to studies, you can use for all ages. Can use for all ages. It is not about the age, about your spermatogenesis, because in males we can reproduce, as long as we can. But if you have got a disease, it makes your reproductive systems worse. So if you have I don't know, a DNA fragmentation or a genetic disease, and you are young, you can't see the benefits. But you don't have a genetic disease, but you are old, you can see the benefit, of course. So no age limit.
Oh, pretty interesting. Okay, and that wraps up our Q&A. Thank you, Dr. Ahmed, for answering our
Thank you.
questions. But before we wrap things up, we have a special announcement for everyone. We're currently working on organizing an in-person workshop focusing on stem cell therapies in IVF, and we hope to invite Dr. Ahmed as well,
Thank
uh,
you.
where part- where participants can have hands-on training and engaging discussions with experts in the field. And we will also have another series of online webinars in collaboration with the European Fertility Society. And as usual, we will share all the details on our IFG LinkedIn page. So follow us and stay tuned. This webinar is recorded as well, and you'll be able to watch this after on our YouTube channel. Dr. Ahmed, maybe you have some final words or some reminders?
Thank you to invite me to this webinar. Thank you very much. I hope we can meet again.
Right. Thank you once again. Thank you everyone for joining us, and we'll see you soon. Goodbye.
Goodbye. It finished, right?
Yeah, Dinus, let us know.